Hello everyone, welcome to this new video. Let's start. In 1968, before 1970s, it was mentioned that the weed-free environment is almost with us with the use of herbicides because herbicides became available that time and the rate of progress is rapid and there is no reason why it should not be possible to get rid of weeds within the next 10 to 20 years. Now, the question is 50 years gone, what happened? Have we got rid of any weeds? Just think about that one with the use of herbicides. I don't think so. So, what should we do? Thing is, just simple message is don't just rely on herbicides. So, with that in mind, today's presentation is about weed biology, mastering weed biology. It is the key to effective weed management. Some of my students, previous postdocs, they suggested that I should first deliver a seminar on weed biology because now this is the hot topic in terms of weed management. People are looking into this type of information, so therefore this is very important. I'm a professor in weed science at the University of Queensland, and I have been working for almost now 25 years in this field. Just background, world's population. We will keep that one, this presentation more related to, you can say, developing countries or low income countries, so that people, and mostly my audience are from these countries, and people can then relate to their situation. So world's population will reach around 8.5, more than 8.5 billion, in fact, by 2030. And agriculture, global agriculture in the 21st century, it has to produce more food and fiber to feed this ever increasing population. So we know that we have to produce more foods for this population. In agriculture, there are several biotic and abiotic factors. Okay, and you know what are those abiotic factors, like your water stress, you call it drought, heat, salinity. And there are several biotic factors, and those biotic can be like insect, disease, weeds that can affect crop production. But weeds among them are the most harmful biotic constraints to agriculture production. Through these two these two papers, we actually tried to explain emerging challenges and opportunities in weed science, and mostly it's in developing countries, but somewhere we also touched in developed countries, and that was about 7 years back. And I think there is more advanced uh, challenges. Sorry, and this one is only recently. And then what are the grand challenges in weed management? And it's mostly related to cropping weeds. And just for your information, weeds are all in different situations. So we can't really just relate to all situations. So you need to really understand. And in this one, mostly I will talk about cropping weeds, but there are weeds in rangelands, there are weeds in environmental sites, also botanical gardens, aquatic weeds. So these are all different. Their ecology, biology is very different. And in future, I will try to bring experts, from those areas. Okay, let's focus again here. Now, globally, weeds cause the highest potential losses, about 30%, and that's compared to other pests, including insects, disease, or nematodes, even rodents, or vertebrates, if we classify them. And if I'm not wrong, even the data suggests that even after incorporating weed management practices, we are actually losing 10% of the total global food production. And 10% is a lot. And I presented in one of that paper that if we have got X amount of grain production worldwide, if weeds are causing 10%, even after we have incorporated those weed management practices, and that 10% is a lot. And if we can say only 5% of that, that will really be very, very you can say, effective, and also it can address hunger or food security in some countries. So weeds, they affect crop production by competing with the crop, we all know, and I have included this one more text so that this presentation is not only for PhD students who already know, or young researchers. Sometimes your master's student, or even uh, bachelor students, students in BSc, for them there is nothing new, or someone comes straight away from 12th class, and if they have not really seen agriculture, for them this is very important to know that weeds compete for resources, and they harbor pests including disease, insects, and some nematodes. Also interfere with crop harvest. Some weeds we know which has got, for example, spines, or some weeds are very green, or they may have high moisture levels at crop harvest, so they can interfere with crop harvest and reduce crop yield. Just in terms of, and this one, I really wish that we should have this data from different countries. And I'm not saying that this all data is very correct, because this was not really taken exactly from fields. It's like average of a few farmers. So, but just so that you understand the importance of weeds. In Australia, for example, that's only around $5 billion, more than $5 billion farmers actually. That's revenue losses to growers in Australia, and that includes weed, that cost uh, in both managing weeds, for example, buying herbicides or using labor, plus yield losses due to weeds. So that total is about 5 billion, and I'm talking about this is mainly in grains, cotton, and main crops, and we are not really looking here rangeland and other situation. In India, it's only in 10 crops of more than $10 billion, and $33 billion in the US. And now you can see that difference between small economy, big economy, or population or land. So you can see there. And with these countries, I can say that 5 billion to $33 billion every year. It's a lot. Okay. Now, manual weeding is still practiced. You can say still practiced, not really very common, but it's still practiced in some countries, and but becoming less common because highways is there, in cities, people are moving there, migration, very hard work. You can see these people bending always, and if they can find better work in cities or somewhere where they don't need to really bend, and they can easily get money, so why would they work here? Also, this weeding is very, very important in terms of timing. You can't really just go and take out weeds when crop is near maturity. You have to take out during that critical weed free period. Migration is happening from rural areas to cities, urban areas. There are some governmental schemes, I know, like in India there were some where people they were getting X amount of hours per month. So if there those schemes are there, why would they go here? So manual bidding is really becoming less common. And sometimes people ask me that, oh, India's population is more than one, or it's now 1.4 billion, and still you get problem with labor. Yes, that's a big problem because of these all different factors. Now we know herbicides are used widely to manage weeds, and I'm not saying that's wrong. It's really good because herbicide use has made agriculture production simple and economical. But there are some concern here. If you see, I know it is from Haryana, India, and I thought this farmer was, giving irrigation or something. Later on, when I went there, he was actually spraying bispyruvate sodium, nominee gold, a herbicide used to control grass weeds in rice, and mostly, this herbicide was promoted for direct seeded rice. And I was surprised to see that this herbicide was really going, and I was standing here, it was going on the face of his wife. It's really a health hazard. Okay, here that's in Sri Lanka when I was at IRI, at the International Rice Research Institute, spraying herbicides, and you can see the nozzle. Nozzle is not really, these nozzles are not for herbicide spray. So how they are used. Also, you can see the situation, how the condition of the field, weeds, stressed. So these are some of the issues. So what are the concerns then if herbicide use has made agriculture very simple and economical? Why then we are talking about it here? What are the concerns? First of all, yes, herbicides, as I mentioned earlier, very good tool, but there is a risk of evolution of resistance, shifts in weed flora, off-site movement of herbicides, limited new modes of action. Now, resistance issues is really becoming big now in even developing countries, or even in countries where people have been using herbicides. And I don't really always say that that's always resistance. Sometimes it's a poor technique, but let's assume here and that's it's all a resistance. If you see globally, more than 500 cases of herbicide resistant, and these are unique herbicide resistant case, and it started around here, that first resistant case, and then until now more than 500. Just give example of glyphosate resistant, and this one Lolium residum. One day I will you know, cover this species, annual ryegrass, very interesting species. And the first glyphosate resistant case was reported from Australia, and now you can see how they are increasing. All these are glyphosate resistant, and I think Vina Fetua, Vina ludoviciana, these were from my lab, and also there was something Johnson grass in Australia. That's also we found glyphosate resistant one. Also, a few years back, I saw this one photo in Tribune. Can't kill weeds, farmers blame herbicides, and really, I don't really understand whether it's really resistant issues or timing, or this. How do we spray herbicides? We need to really. I feel our extension people need to be very active or proactive, and change these nozzles, and make sure that herbicides are used effectively, correctly, at the correct doses, timing, all those things. Okay, but there are some these issues are real. These are real issues. I'm not saying that resistance is not happening, but also there is shift in weed flora now, hard to control weeds, or your herbicides got rid of one weed and then there is another one dominated that system. Also off-site movement, especially if we have fish farming nearby, or there are rivers. So that's really, and in Australia definitely it's an issue. A new mode of action. Now I think there is some good news, some new modes of actions coming, but last 20 years there were not many. Okay, but I don't think that in the next 10 years there will be more than 2 3 ones. It's very limited. And the first case of, as I mentioned, glyphosate resistant ryegrass in 1996, and the same thing I know my teachers, they reported from Haryana Agriculture University, herbicide resistant canary grass 95 in India, and that's called Flaris or Flaris minor, or Gulli Danda, whatever you call it. And since then, several populations have developed. Okay, and I don't really think that we want to lead this list of different countries in herbicide resistant weeds. Now, the challenges for scientists mainly is to develop these three: innovative, economical, and sustainable integrated weed management. Integrated weed management is not really a new term, but now we are talking about that should be economical. Not really, you can't really just have in research 6 different methods and you propose that one. That may not be economical. Should be innovative, bring something new there. Okay, and sustainable. It can sustain in a long time. Can't really just go, and I still see in some countries their treatments, everything includes followed by hand weeding. I think we need to go away from that hand weeding and bring some mechanical weed management methods. At this moment, most efforts in most of these countries are on evaluating herbicides. I really surprised when I go in some countries, and I can name here, South Asian countries, including India. People always talk about, oh, what is the new herbicide for this for this weed? I am not really in private companies. That's not really scientist work. Okay, I feel scientists or researchers should not really just go and evaluate different herbicides. I don't think that there is any science there. Already private companies are bringing these herbicides. They already know, they have already evaluated. Our focus should be like how can we keep those herbicides sustainable for long period of time, okay, in long term for long-term management of these weeds so that we don't really break these herbicides and lose them. Okay, also we need to really understand that, okay, if farmers are using these herbicides in stressed conditions, what should we do uh, to improve that their control. So that type of information we should need, not really just evaluating different herbicides. And there is an urgent need now to develop ecological based integrated weed management programs. I will explain in a slide what it's mean and why people always get confused. And to develop effective and sustainable these programs, a better understanding of weed biology is important. Weed biology is not really just simple term. It's really a broad term. I can't really cover everything here. I will cover maybe 10-20% of the total weed biology work. But its understanding is very important. And in my lab, personally, I don't really just want to go and kill a weed. I first want to understand about that weed, how it germinates, how it grows, okay, and where are the weak points in this weed, okay. Harnessing that knowledge will spearhead ability to implement integrated weed management, and that will rely on precision rather than just herbicide dependence. So we need to really reduce our dependence on herbicides. Now, just want to cover in two slides why is weed science lagging behind, and I'm comparing with other crop protection fields like entomology or pathology. Why we are lagging behind? Um, I think I just give this example, like for example, in India I covered earlier, or it can be in Asia or in Africa. So uh, I will cover mostly these two, Asia and that's both South Asia and Southeast Asia as well as in Africa. So weed control, if you know, was a minor part of agronomy and plant physiology. And I feel really now also, it's still in some countries, it's only a minor part. I don't really see any weed scientist. And most of these in African countries, in Southeast Asia, I don't really see a weed scientist there. So they are all agronomy, but it was minor part of agronomy or plant physiology until 1950s when herbicides became widely available later on. And until now, emphasis has been always in weed science on how to control a weed rather than understanding its knowledge of the cause or how these weeds germinate. And this is really in contrast to plant pathology and entomology. I know in these fields, mostly studies first centered on what is they identify the cause of disease or understand that if there is an insect, they try to understand its life cycle and what its host and damage so that from that information a method of control would grow, okay. And they have made significant progress towards the development, assessment, and implementation of integrated pest and disease management, but discipline of weed science continues to lack. Now I will come back to my that first slide point. Weed-free environment is almost with us with the use of herbicides. So what happened? Again, you try to think there. Because of this one, no weed species has been eradicated by the use of, I'm talking about only herbicides. When you use only herbicides, just rely on herbicides, no weed species have been eradicated. Okay. But a number of troublesome weeds have increased. So therefore, herbicide use, I again emphasize this point, which I covered in the last slide, that there is a need to change the thinking of farmers away from a yes, provided that, to a no unless attitude for herbicide spray. Now, just in one slide, and I think many people can relate this one, and I know, especially in India, also in Bangladesh, Pakistan, they will relate that one, and they always are criticized if someone is studying, weed biology. Why does not weed biology get importance? Just in simple term, first of all, people look always for quick fix, and I think I blame always our extension people also. They always look for herbicides rather than understanding about those. I can understand their situation, but they need to really understand not all weed scientists should work or will have that knowledge of different herbicides, because weed science is not about just herbicides. Okay, so it's a long-term weed biology studies, a long-term studies. It's not really just go there, apply 10 herbicides and find that, oh, X herbicide is working. Also, weed biology studies, they do not create. Now you need to understand always why it's criticized, because herbicides provide you a tool, okay, but these weed biology studies, they don't provide new products or any tool. But what they do, they provide a concept for weed management. That's what you need to understand. A concept of weed management that can be successfully used in making informed decision and in the selection of control tactics for researchers, growers, and industry. And I think after that one, it becomes our means weed researchers' responsibility how you convey that information. And I'm glad in Australia now we get $1 million project just to study biology of weed species, because our growers also want to know now, oh, how long it survives in the soil, and when do uh, the germinate, all those type of questions. That's very important, and that's what I think we need to really start doing in these countries. Okay, I will give selected quotations when I discussed in the last 4, 5, 10 years about weed biology. That, oh, why don't you give some work to students? Just their quotation. If we include experiments on weed biology or basic weed research, students' synopsis will be rejected. But I am glad that now some universities, and I know in Punjab Agriculture University and in Haryana Agriculture University, I co-supervised students, and they are trying to study this basic research. But I think in most universities, still it's not really fashion there. We work for farmers. That's another quotation. We work for farmers, and weed biology information does not help them. Okay, is it a really comment? We will never get approval from our head. I think you should change those heads if they don't really talk about or think of for a long period of time. And that's why this is I got it from Central India, from I think Directorate of Weed Research. It's in Jabalpur, and I always give, and I'm glad that in the world there's only one center, one directorate on weed science. It's not really nowhere, and where you can find people from they have got different disciplines within weed science. Someone is on disease, okay, someone on their use, someone on chemicals, and also someone on herbicide residue. So to me that's very important, weed knowledge to benefit everyone. That's really very important notation. Now, as I mentioned earlier, weed biology is a broad term. I can't really cover everything here, but some of these ones are genetic diversity, seed physiology like dormancy, how long seed is dormant, how long they persist in the soil. Weed physiology also, chemical and physical process involved in those weeds. Emergence pattern, when do they emerge in a year time. Their reproductive biology, phenotypic plasticity, do they show any plasticity in terms of maybe stress or crop competition, whatever we are talking about. Competitiveness and its allelopathy as well as parasitism. Fitness cost, if there are herbicide resistant weeds, is there any fitness cost in that particular population? So this is all there included in weed biology. We will not cover everything. Let's see a few we will cover and their importance. First of all, genetic diversity. It's increasing knowledge of that one. Genetic diversity means there are plants or Yeah, there are plants, they differ. If you collect them from different places, they may have very different genetic uh, makeup. So increasing knowledge of that one may answer few questions, and that you will have better understanding once you know the questions what we are trying to answer. Is the selections of weeds being imposed by management, whether that diversity, whether that was due to different management, and management I mean like different herbicides or different your tillage practices, whatever you say, or when people are talking about direct seeded rice versus transplanted rice, whether it's imposed by management, and is the intensity of selection pressure imposed by agriculture practices more powerful than by natural ecosystem? Okay, so you need to differentiate between these two, agriculture practices or natural ecosystem. Have weeds evolved and keep evolving in response to agriculture practices? So again, it's the same thing, management. So agriculture practices, we keep changing. Have these weeds evolved? And recently, I attended a conference, and in Western Australia, they found that wild radish plants, it's usually, it's around 4 months duration, that weed, but they found that some plants actually they were selecting for early seed shattering within, like, 2 months time. So that's how they evolved, because they keep growing. You will see, like, even if there are 1 million plants, one plant will behave differently, and if we allow that plant to survive, that will produce more seeds next year. Out of those seeds will behave the same way, and that's how populations increases. And is the success of management programs associated with this to genetic diversity of weeds? So these all questions can be answered if we study the genetic diversity of weeds in your area. Now I will talk about seed bank in the next slide. What's that one? But see, people are always talking about different tillage system. I just is from 20 years back. People say that, oh, just example, this weed is less in no-till and more in conventional tillage. My question is, that's okay, that's all right, but our question should be, and the same thing here. I know that in this particular system, it's about, just say, 1500 plants. In this one, it's only like, just say, 300 plants. My question is not really that it got less. Okay, what I'm trying to understand is what's happening with this gap. Those 1200 plants, what happened to those ones? Why they didn't germinate? If they are dormant, they are not really good. They will come in the next season and it will really create more problem. But if they are dying somewhere, because for example, if it is a no-till system, most seeds remain on the soil surface. And if they desiccate due to unfavorable weather conditions, that's good thing. So, but we need to really understand. And before that one, how will you understand? First, you need to understand the seed bank. Okay. Some people say that, okay, what is this seed bank? It's only that in the soil. No, just think that it's a soil, like your maybe bank or pool of something. How the seeds are coming? First of all, seed rain from your plants, they produce seeds, or seed dispersal through water, wind, animal, machinery, whatever. Seed comes in the soil. So these are some of the seeds there. Now, some seeds out of these ones, they decay due to maybe pathogens or any other environments. Okay, although it's good, so it means we know that we are actually finishing these seed bank. Some are taken by predators, and ants are the main one, birds, mice, insects. Some are these ones. That's also good if they have eaten them. If they are just storing, that's not really good. So if they are eaten, that's always good. So still we are finishing. Some will germinate and die. That's again good. Some will germinate and establish. That's also good in terms of, because we know, we can see them. There are plants they establish. We can see them from our eyes, and we can go and do some management practices, okay, and kill them. But the real problem are these ones. Try to think, what are these ones? Give you some time. Okay, these are the problem. These are dormant seeds, and I think that's what we need to try to understand. So for me, weeds are just symptom of the problem. The real problem is weed seed bank. Okay, so therefore, focused research should be in reducing that seed bank. What are the practices which will help reduce seed bank? And I always ask, just suggest to my students, you try to find something, which can help releasing weed seeds from dormancy. Either can be any novel chemicals. People tried, gibberellic acid, but that's very expensive. You can't really use in the field. Sometimes you use potassium nitrate and all those things, but you also need to see that you are not increasing that chemical in the field, and they are very effective. Problem is people end up using just doing this type of work in pots or in labs. When you go to the field, they don't really work. So we need to evaluate something which will help them. Okay. Growers would benefit from any practice, whatever can cut weed seed input and increase seed losses. Now, I think even when we publish papers, we always ask that, suggest that you should give weed seed data, not only just to plant biomass and yield, because this is the one, what your treatments in the field, how much they are adding, seed to the field. So that's why it's very important. Now let's cover a few of these ones. First of all, see what are the factors. What say about seed dormancy or germination? What are the factors that can affect dormancy? Light, we know already. Some of that one that light can affect. I'll cover a little bit about that one. Temperature is another factor which can affect. Whether it's constant or fluctuating. Mostly on the surface. Less fluctuating compared to your buried. Deep more fluctuating, sorry, compared to seeds buried at depth. Burial depth also can affect emergence. Seed scarification and fire. Fire is also happening in some areas. And soil moisture can affect crop residue. Depth, flooding depth and duration, it's more in rice systems. Soil salinity, especially in coastal areas, pH and nutrients, mainly nitrogen, they can affect. So information on germination response to these factors I'm talking about, it can help develop effective weed management programs, either through spressing. So you can spress using, for example, crop residue, or increase this germination, for example, by fire or scarification, and then you control them. Okay, that's why it's very important. See this one, first try to understand light. This is our soil profile, seeds on the surface, they get exposed to light, but mind you, like I think mostly seeds in the top 5 millimeter or half centimeter get direct light there. Others, they don't really get light, so that's why they remain in the dark. So you need to understand weeds respond differently to light and darkness. If they are buried here, they will behave differently. But that also depends on species. So responses are always species specific. Some germinate equally in light and dark, doesn't matter. Yeah, it should not be that deep because it may not have energy to come to the surface. But if they are here or there, it does not really make difference. Sometimes light stimulates germination. So you will still get, maybe for example, 90% germination here, you get 70%. So still you are getting high germination for buried seeds, but light will stimulate, so there will be more. But for some weeds, absolute light requirement for germination, like leptoclova or something. If they are buried here, you won't get any germination, and that's we call it always positively photoblastic. And light for some weed species that inhibit germination. That was the first species I found during my PhD 20 years back, and I still remember that work. In that one, light inhibit germination. I will cover that one. Okay, so that I will show you a graph, but before that, so that you understand some of these, you know some of these weeds. This is rice flat sedge or cypress area. This is the sedge, milk thistle, common saw thistle, Sonchus oleracea, marshmallow, Malva parviflora. It's increasing in some nodal system. This is Leptoclova chinensis, Chinese sprangletop, and that's what people were complaining about in direct seeded rice when they were applying bispyruvic sodium. Now, if you see what I mean by species specific, you can see easily later on, if you don't really get time now, you can see these names, Chinese sprangletop, and these are all there. So what's that mean? You see here light and dark, that's germination on y-axis, and these are different weed species. See, some of these weeds, they don't have any germination in dark. So it means they have absolute requirement for light. If you bury them in the soil, even at 1 centimeter, they may not emerge. Okay. Some of them, these ones, there are different, weed species where germination is happening in both dark and light conditions when they are buried at shallow burial, even in light, but light actually stimulates germination. And there are some where there is no difference at all, and these are usually hard seededness. Can say marshmallow, wild turnip, and I'm talking about shallow, 4 centimeter, 5 centimeter. I'm not really talking about 20 centimeter depth. From there, it can't really germinate. So this is dark. Okay, how we do this type of experiment in the lab? We just wrap in 3 layers of aluminum foil, and that's what where light can't penetrate, and then check germination, and then we actually confirm this through seed burial depth experiment. Now you have now better understanding of what I mean. Like most species, they require light to be released from dormancy because they are small seeded ones, and light's requirement can be actually satisfied even after a brief exposure to light. For example, soil tillage, if you're doing just a fraction of a second, and after that seeds buried again, but it has already satisfied its light. So it means a species showing preference for light in germination always has potential to be prevalent in no-till system. Why is that? Because most of wheat seeds remain on the soil surface after cultivation or after planting in no-till system because we are not burying them. So if they have preference for light, they will become problem there. But some seeds which require light, you can inhibit them if they are buried deep or don't really bring them back, and they will remain dormant there in the soil if they are already in the soil because uh, you are not doing cultivation, so you are not bringing them, and then they eventually lost to predation and decay. So dormancy, how dormancy can be used to manage weed population, that's how you need to really use this dormancy thing. Night cultivation was used in some countries, not for this purpose, for reducing weeds, but because in some countries, especially in high income countries, their land size is very big, their planting window is really very narrow. So if they are cultivating, they were doing in the night. And we found that in night cultivation, uh, some species there, because they had requirement for light, fat hen or Chenopodium album, Bathu, and this one is Amaranthus. And their actually germination was reduced if when people followed a nighttime cultivation. So that can be used. Not people are using this one just to manage, but they are cultivating during night time just to cover their whole area. So light acts as a depth indicator for seeds and prevent germination of buried seeds, promote germination of only those which are near the surface. So absence of a germination response to light suggests the seeds may germinate even when buried or after canopy closure, because when crop has closed its canopy, still there is dark. So absence of that response suggests that these species may germinate. And deeply buried seeds will be fatal, that germination, if unable to reach the surface. So it means it can germinate in dark, and if it is, for example, if it is buried at 10 centimeter, it will start germinating, but it may not be able to reach the soil surface because seeds may be too small and may not have enough seed reserves or food in simple terms, and it will die. So while seedlings emerging after crop canopy closure are likely to experience great competition for light, water, nutrients, all those resources, and may result in less crop yield loss and weed seed production. That's very important. And on another point, if seeds need light to germinate and have low dormancy, I'm talking about, for example, Leptocloa chinensis or Chinese sprangletop, milk thistle, they don't have dormancy. So stale seed bed can reduce because seeds are mostly on the surface. You know, these are seeds there. You irrigate them and they germinate. And some people they are using for, where uh, herbicide resistant rice is not available, and I will cover that in next few weeks. Uh, there they are using this stale seed bed. And what is a stale seed bed? Promoting weed seed germination by light irrigation before land preparation and kill those seedlings by non-selective herbicides. And again, if you have allowed that one, some apply herbicides, some they do tillage. But again, tillage will stimulate more emergence. So you need to really understand whether you can afford that emergence. Two things: if there will be more emergence, you are actually reducing or exhausting your seed bank. And if you don't do tillage, you go with the herbicides, then those weed seeds may germinate in your crop period. All right, I hope this light thing was all clear. Next one is temperature. Temperature, we know in past people were saying winter weeds, summer weeds, but now really changing this one. That's why I think it's very important for you to understand. This information helps us to understand the spread in different regions or seasons if we know effect of temperature. So this is just an example, one weed. You don't really need to understand or know the weed. This is just an example. It's a feathertop rhodes grass, mainly a summer weed in our situation, but in the last 4 or 5 years, I've been actually reporting this one even in winter seasons. So what we did, and it's germination. There are two different things you need to know: germination and growth. Okay, I'm here talking about germination. So when we germinated that at different temperatures. This is like y-axis. This is some y-axis is germination, x-axis temperature, and these are 12 hours. 12 hours during daytime, 12 hours during nighttime. So this is like your winter temperature, 15.5 to your maximum 35 during daytime, 25 during nighttime. If you see, these are different lines are different populations. So it's not really that all populations will behave similarly. So some population had maximum emergence at the lowest temperature, and others had here, So here 25, 15. So you need to really understand this one. So seeds of feathertop germinated at all tested temperature, and what it indicates, what it's mean if we have that information. It indicates its ability to germinate throughout spring, summer, and autumn in Queensland, in our region in Australia. Same thing in your region. If you know that what are your winter temperature, what are your summer or extreme temperature, you can test this one, and then you can relate that one. And that will also help to understand the spread in different regions. I will cover that later on. I actually warned that time and people didn't listen to me. And now we see this weed in winter crops, in wheat crop. And I'll cover that later on. And what's that mean if they, because they are in summer weeds, if they germinate in spring, which comes here after winter, that would make weed control difficult as winter cereal crops are still growing in the field. So if they germinate in winter, it's really difficult, which is happening now. After 3 months of winter, when we get spring, it even if that some of these populations, this is some spring temperature, if they germinate then, and your winter crop is still there and it start opening its canopy because it's near maturity, that will become problem. There's you will have these plants still in the field in winter crops, and you are still harvesting that one. So if people want to go later on and read more about that one and what its implication, this is the paper my student published on this one in Weed Science. Now, seed scarification. First of all, you need to understand scarification is like, in simple terms, cutting or softening that one. So seed coat actually can impose dormancy. There are some plants like, people are talking about marshmallow. This one is Mimosa invisa. Some areas they call it touch me not, or sensitive, giant sensitive plant or sensitive plant. So some of them, they hard seeds they require. They have hard seeds. If you see some of these ones, like it is I think wild jute, that's marshmallow, Milokia red, weed there, or chocolate weed they call it. These are two species of sensitive plants. Sida is there, Urina lobata. So if you see, we did not do any scarification. And then when we scarified either hot water or physical scarification or chemical scarification with sulfuric acid, you can see germination. So see the comparison. So very low versus this one. So you need to really understand why, when now you know that why we get germination of these hard seeded ones over long period of time. And from this one, I can tell you these type of weed species will always last long in the field. They may last for 10, 15, 20 years. Okay, their seed bank persistence. So these hard seeds, they require scarification to enhance germination. So what are the mechanisms? Just try to think about without looking into this slide. Think about that if you are in your situation, whether you are in rangeland or cropping weeds, what are the mechanisms that will increase? What are the operations that will increase breakdown of seed count and that will increase your germination of these weeds? First of all, try to think about changes in temperature and moisture regimes, where in your profile, in soil profile, maximum fluctuation, because that will increase more breakdown of seed coat, and maximum fluctuation is on the surface. So it means which system is better for seed bank depletion. I'm giving just example, no-till or conventional, where we just cultivate. So both of these ones is no-till system. Why? Because fluctuation always reduces when we go deeper in the soil. Same thing here, seed bank depletion will be more because seeds are on the surface. So it will be more in no-till system. Also, breakdown can be because of microbial and fungi attack. You need to understand where is that one, whether you have got a field without crop residue, with crop residue, abrasion by soil particles, that's very minimal tillage. But if people are cultivating very thoroughly using heavy machineries and cultivating 10 times in a year, that can affect. Also soil, acidic soils, that may also help breakdown of seed coat. Also, if you have got animals in the field that's trampling by hoofed animals like sheep or cattle, that can also increase breakdown of seed coat. It means more germination. Also, passage through animals' digestive tract. We know depending on weed species, some of them like acacia can um, animal can digest some of them, but most of them can come out, and it takes up to 7 days to come out completely. So you need to really understand it. Also fire, if you see in some situation, either it's deliberately or somewhere, if you see fire in forest, and you will see that some of these hard seededness species will germinate more after that fire. I know in Laos, uh, we still they have slash and burning system. So they slash some forest area and then burn it and then plant crops. In their situation also, after fire they get germination of these some of these weeds. Next one is seed burial depth. First of all, you need to understand how is these seeds buried in at different depths. Whatever you do, that will affect vertical distribution. Either you are cultivating or shallow cultivation, planting your crop, or doing hand weeding. Whatever you are doing, you are disturbing the soil, and soil disturbance will affect vertical weed seed distribution. Weed seeds is on the surface or buried. Okay, and that. So now just try to understand this one. Tillage affects vertical seed distribution. Try to understand this graph. For example, this is annual ryegrass seeds, and it's 20 years back. I got it. I put these seeds in the on the soil surface, and it was covered with this powder. Uh, and this is the same type of one. If you see, sometimes in our watch, we see during night time, that's a reflective one. So that powder, I coated these ryegrass seeds, placed on the soil surface, planted crops or cultivated, and then took samples from 0 to 1, 1 to 5, and 5 to 10 centimeter, and then counted these seeds under an ultraviolet light. If you see here, conventional tillage system, and it was tilling up to 10 centimeter, and no-till, because of different system, how deep it can go. What we found that no-till system, because we use this tine system, and tine and disc creates less disturbance. Tines creates more disturbance. So we used tine system. So what happened here, no-till system, mostly because it's low disturbing, it was only planting that crop, so it retained most seeds on the surface. If you see, 55% seeds are on the surface, 0 to 1 centimeter, from 1 to 5 around 30, and 5 to 10 only 10. But conventional one, if you see, it was mostly in this in this depth, middle one, 1 to 5 centimeter, or 5 to 10. On the top one, it's opposite to a no-till system, only like 5% seeds were there. So conventional tillage distributes uniformly because you are cultivating and mixing those seeds. But in no-till, because you are just planting your crop, so it remains on the surface, mostly in the surface layer. So once you understand that thing, so then you can easily say that this distribution has the potential to affect seedling emergence. Now you know where are your weed seeds in this system. So weed, and it will affect weed population dynamics. For example, seeds are here, you will get light, more temperature fluctuation. Here you may get better moisture, more predation here, less predation here, more decay here. So there are all different factors will affect where are your seeds, and that's how you can say that seeds present near the surface, they germinate and emerge earlier than deep buried, because they are buried deep, they may not have light, and here they may have favorable conditions, they can stay there longer because they don't really have. So you need to really understand both way. It can go in both way. You need to understand depending on weed species. And again, if someone wants to see this this particular paper, you can go there, influence of tillage system on vertical distribution and persistence. I covered also how where seeds were more persisted. Now seed burial depth, if we now you understand what seed burial depth, we have done, I think my lab has done work on more about 100 weed species where we have done this type of work. This is again all from my lab, but mostly this is, I think this is all from my work from the International Rice Research Institute. So optimum burial depth, what's that mean? Maximum number of sown seeds can produce. So just an example, maximum for most of them on the surface. So mostly surface, this is x-axis, its seed burial depth, and y-axis emergence. If you see in most of them, Amaranthus viridis, Digitaria, longifolia, see both of them, they are behaving very differently. No more emergence after this 1 centimeter, but in this Digitaria ciliaris, both are the same genus. It's behaving very differently. Echinocloa colonna, elucid, mimosa. If you see here, up to 8 centimeter, it can emerge. Mimosa invisa, and then these are Estraceae, Melokia concatenata. Interesting one is Leptocloa. If you understand this one, this is a Chinese sprangletop, and that's what I mentioned in the first slide, that it actually has absolute light requirement. So it's germinated only when it was on the surface. As soon as I buried that one at 0.5 centimeter, no emergence occurred. So this type of information is important, and now we know what is their optimum burial depth. But what's important is we should really know what's the maximum depth from where they can't really emerge, so that if someone has to do tillage, they know that I have to bury these seeds deeper than X depth. So generally greater seedling emergence from the surface, but species response differ in their response to depth. And also if you see, this is the seed size. That's really very important. These are some weeds. And if you see here, if soybean seed versus some of your weed seeds, this is ryegrass seed, sesbania seed. Here in Australia, it's a weed. It's a liver seed grass. Okay, that's a windmill grass. Some of these ones, ageratum. So see the size versus this one. So that's why in our situation sometimes farmers, they plant their crops very deep, so that it remains always dry on the soil surface. Most of that even 4 centimeter, and some of these weeds can't emerge, and their crop can emerge. You won't believe one farmer I met, and he planted his chickpea at 15 centimeter depth. Now, first you need to understand, in Australia we have got dryland agriculture, so all depend on rainfall. So what's that mean? 15 centimeter. I was worried that that would not emerge, but what he did, planted in furrows, so it was around 15 from the top, and top 5 centimeter was totally dry. And we know that 90% of our weed seeds, they germinate from the top 2 centimeters. So it means there was no moisture in the top layer, and is actually crop was able to emerge from that depth. and he did not have any weeds problem. So that's how some people are using in dryland agriculture that information. So greater emergence from seeds on or near the surface. It means, as we mentioned in that discussed in the light experiment or light slide, that no-till practices would enhance emergence of these small seeded species, as a large proportion of the seed bank remains on the surface after planting. Okay. With few exceptions, emergence from the weed seed bank occurs only from the upper few centimeters soil layer. Okay. Weeds could be managed by burying, and that's what some of our farmers are doing here. When they have got concentration of weed seeds on the soil surface, they go and bury these seeds below the maximum depth uh, with deep tillage, and subsequently use shallow tillage so that they don't really bring back the seed bank on the soil surface. So greater knowledge on the impact of seed burial depth on target species could improve weed control and reduce reliance on other options. Now I will cover the last one is crop residue in these ones, factors affecting this one. Just for your information, this is a situation, for example, this is in India on the right hand side. In Australia, uh, it's called windrow burning. Sometimes they collect all together. It's really very heavy, thick, and then they burn it, this one. And some of the weed seeds are also there. But that's not my purpose here. I'm talking about crop residue as a physical barrier. That's very common in Australia, especially in sorghum. We get very high residue and that remain on the surface because sorghum, they keep at least 40-50 centimeter of residue there, and over time it decompose and make a layer here. So provides a physical barrier to emerging weeds. So it means it can reduce emergence but also delay emergence if you get emergence from that deep. And but the impact depends on the quantity of the residue. As I mentioned earlier, sometimes we get only 1 ton per hectare yield. That may not have that enough residue. Sometimes positions of your weeds related to that's also depend on whether you had already residue and then your weeds seed shattered, so it means it's on the surface, it will emerge faster, or your weed seed shattered and then farmer harvested their crop and this one actually covered the weed seed, so that will also affect. Also allelopathic potential of the residue. We know that sorghum has got high allelopathic potential, but some crops may not have. Within a crop, some varieties have got high allelopathic potential. So the impact of crop residue on weed emergence will depend on um, these 3-4 things. Also, people are now talking about cover crops in some situations. They have also, crop residue. Rather than they take their cover crops are terminated either by spraying herbicides, or sometimes in some situations they incorporate using tillage, or in some other cases it's mowing and leaving that residue on the surface. Depending on plant species, for example, some brassicas, they have got high allelochemicals, so that can also actually suppress weeds, and this is being used now in the US. It's, I think, all universities are really doing routine work. In Australia we are trying, but our challenge is very different. Our growers need to know whether they want to grow cover crops with that limited moisture because we just depend on rainfall, or they want to grow their main crop. So if you see crop residue, just giving example, again it's species specific. If you see x-axis, it's residue amount, and I think it's only wheat crop residue, 0 to 8 tons, depending on, in some situations we get only 1 ton. Where there is irrigation, you may get something 8 tons. On y-axis emergence. So these are three different weeds. You don't need to remember which are these ones. What I want to emphasize is the response is specific. So generally, weed emergence decreases with an increase in residue biomass. If you see all of these three, African turnip weed is like this one is sorghum residue. So emergence decreased linearly if you see there. For barnyard grass, which is called jungle rice, we call it ownless barnyard grass. It's jungle rice in the US, or samwak in some countries. It's Echinoclea colonna. it's rice residue used, that's emergence decreased exponentially. So that's linearly, exponentially, and here again exponentially. It's windmill grass, but sorghum residue. Okay, sorry, I think I mentioned wheat residue. These all different ones, sorghum and rice residue. Here, emergence decreased exponentially. If you see here, so see, different weed species will respond differently. So the response varies among species. Some can increase at low rates. I have seen in some when you have got only 1 ton, their emergence is actually increases. Why? Because they have got better seed soil moisture contact, and then it can reduce. Now, people in some countries, especially from South Asia, can relate this one. Weed suppression is attributed. This picture I'm talking about. So residue burning is really common, and especially in rice, smog. It's happening. Environmental issues are there. So weed suppression is attributed to shading, as I showed here, temperature, because there is less fluctuation, obstruction, or allelopathy. And usually, as you saw in the previous slides, quantities are greater than those normally found in fields, but the use of mulches may become relevant due to concerns about depletion of soil organic matter and environmental pollution due to burning. So therefore, there is a need to integrate mulch retention with other management options. Again, I want to emphasize this I reported that earlier, but again, weed biology studies do not create new management products like herbicides. What they do, they provide a concept for weed management that can be successfully used in making informed decisions in the selection of control tactics for researchers, growers, and industries. So I hope now